Hello everyone, how are you today? I did for the record press the record button today, so there are not to forget. Last lecture I forgot pressing the record button, so for those of you who intended to review the lecture afterwards, they've been in trouble because there is no recording of the last lecture because of my stupidity. So I apologize for that. And so last lecture, we actually this chapter is about technology. So the purpose of this chapter is to have discuss the principle, although it's a technical, but unless you understand the basic principle behind the technology, you will never be able to understand those progress of all this technology, which is actually, in a sense, the core, the essence of the popularity and the importance of the life science in these days, due to actually this type of the technical advances making your everyday lives something different, and also which is also scary is that it's going to actually change a lot of your future lives, or due to, thanks to, or because of, depending on how you view these technical advances. So last lecture we went over first of actually the starting point, the restriction endonuclease. It was the molecular scissor. I compared that restriction enzyme as a molecular scissors because thanks to the discovery of those enzymes, there are a whole bunch of array of enzymes that recognize specific sets of DNA sequences and then only bind to that area of the DNA and then cut them in very unique way. And the importance of such behavior was so that we could human, because they are all derived from bacteria for their own defense mechanism. But anyhow, we took advantage of their defense system to our advantage in using, like, wherever, whichever, conveniently, if we want to cut certain area of the DNA segment, then we could use different combination of the restriction enzyme so that we could isolate those DNA fragment to clone. So like DNA cloning, the gene cloning was the starting point of gene cloning was without the restriction enzyme, we were not able to start those things actually. So as I mentioned last lecture, the so-called the quote, playing God, because the initial attempt was actually try to isolate the human insulin gene, and then put that gene segment into certain type of a vector, and then put them in the bacteria so that bacteria can produce in large amount of the insulin, so that we can nowadays we can enjoy the abundance and the cheap availability of the insulin thanks to them. But then actually the initial pioneering scientists back then, the scientists at Harvard and also some institute in California, really played this very carefully because of they were also concerned of what if those general public blamed us. And what I do, how dare you play God? And how can you manipulate the gene that those genes were something that God gave you? Okay, you don't have any permission to manipulate them, type of thing. So anyway, it went relatively smooth. So because of that, we were able to utilize those things, whether that's good or not, depending on, yeah, it's all totally up to your individual opinion. But anyway, so those restriction enzyme, and also very simplistic plain technique, but also reliable technique of separating those DNA fragments using gel electrophoresis. Okay, that we went over such techniques. So using such primitive looking technique, but still reliable, so that everybody used them in the laboratory. And so you can isolate, purify those nucleic acid and even protein by their different sizes because utilizing different mobility in a specific medium. And now, so we also try to look at another very important technique developed, also this plus U, DNA sequencing. It is an initial conventional way of getting this DNA sequence technique. It's called Sanger because the inventor was Frederick Sanger. As you can see, this person won Nobel Prize twice even. Very impressive. But during his school, he was not a very good student, at least in terms of the GPA, grade point average wise. He got flunked out in many courses. So what can he learn from that? The GPA, your grade point average is just meaningless. Okay? So just because you, by the way, I'm really sorry that I couldn't still finish your midterm exam grading yet. So definitely no later than next Monday I will be before, no later than, I promise. Next Monday I will get everything ready. My sincere apology, but that's me. But anyway, so this scientist was not really regarded as a very good student, but still was able to generate this really, really remarkable and pioneering results in science footage. So anyway, the principle of this technology is, let's think about it. Let's say this is one length of a DNA fragment, double-stranded DNA fragment, let's say, imagine. And if you, for some reason, out of curiosity, if you want to identify each DNA sequence, let's say this DNA fragment is like 100 nucleotide long. If you want to identify the order of all those 100 different nucleotides, that's what we call DNA sequencing. So this DNA, the upper fragment probably will run, and this end is a 5' end, and this end is a 3' end. And if that is the case, the complementary strand in this double strand is here. This will be 5' end, and this will be 3' end. That's always the rule, right? So if you want to identify the DNA sequence of this, like starting from here, this 1, 2, 3, 4, 5, 6, all this nucleotide in sequence. Well, the beauty of this DNA sequencing is you don't really have to identify both the strand, right? As long as you get the one DNA sequence, and the other one is automatically given because the complementary rule exists. So you don't really worry about how. How do you? What is your individual approach? In your opinion, what would you do if you had to? If you had to invent the technique by which this DNA sequence was identified. Like human instinct is probably, okay, individual DNA sequence. Then I want to break this whole fragment into single pieces and then try to somehow get the order and then identify. And actually there is that type of method also exists. We call this a chemical method of DNA sequencing. This is the old way, although in these days some people need to use such method, but compared with this Sanger method, it is by far more inconvenient and also there's some troublesome. So that's why this one get by far the vast majority of popularity. So the key point is using DNA polymerase to get the sequence information. DNA polymerase is what kind of enzyme is it? DNA polymerase is an enzyme which synthesizes the nucleotide. But how can you? Your instinct usually would probably tell you that you want to know this sequence, you want to chop this individual DNA nucleus so that individually you can identify piece by piece. It's probably most of you, including me, would have approached in this way. But this gentleman and scientists think totally the other way around and try to identify this sequence by during the process of actual synthesis. So that's the key of this, the so-called DNA, Sanger DNA sequencing method. And another name is also usually, actually more official name for this DNA sequencing is this dideoxy chain termination method. That name provides a lot of a clue about this principle of this technology. What? Dideoxy. What is this? And another thing is a chain termination. What is this? So dideoxy. Try to compare these two nucleotides. One is original, regular DNA nucleotide, and the other one is unusual nucleotide. You can easily see the difference because they were compared in colors. One is this and the other one is this. So the one in the bottom is called dideoxy. Dideoxy. The original DNA nucleotide is a deoxyribonucleic acid. The reason is because deoxy is here. This position, oxygen is missing. That's why that is the reason why this nucleotide, which is the regular member of the DNA, is called deoxyribonucleic acid. But here, in addition to this position, deoxygenation, this position is also oxygen is missing. That's why it is called double deoxynucleotide. If you include, during the DNA synthesis procedure, if you include, you don't have to, you cannot provide all of those so-called, you have to be able to understand this terminology, DNTP. I've once mentioned this DNTP, but what is DNTP? DNTP means not T. The N is A, G, C, T, all four. So DNTP means DATP, DGTP, DCTP, DTTP, everything. All four different kinds of, here deoxy. So DNTP means the regular ingredient, the nucleotide that are used and needed for DNA synthesis. Because DNTP, each individual DNTP here, this is one of those DNTP. And as I shown you the other day, during the synthesis, this 2-phosphate is clipped up, and only this part will be incorporated into the DNA chain. You don't remember the figure that showed you why this DNA synthesis utilized DNTP instead of the NMP, monophosphate? I didn't mention it? Yes, I did, I think. It was there. So because of this energy requirement, I don't need to go over that again anyway. So this regular nucleotide, so you have to have, for any DNA synthesis to get going, these 4 different types of nucleotide in 1 to 3 triphosphate form is absolutely required one. However, what if during that reaction, what if you also add small amount of this double dideoxy DNTP, small amount, very small amount. What's going to happen? Any idea? The clue is the name of this sequencing. What's it called? Dideoxy chain termination reaction. What do you think it will happen if this added dideoxynucleotide? There will be some occasions, randomly, according to this nucleotide will be inserted according to the original sequence of the template. Because DNA polymerization reaction is simply putting the complementary nucleotide to the sequence of a template, adding up, right? Picking up. So while majority of these regular deoxynucleotides will be inserted according to the complementarity rule, but then occasionally, because it is also present, randomly, this polymerase might pick up this one and then try to match. Because these two different nucleotides, you cannot really distinguish the difference because structurally they are very similar. The only difference is this versus that. So DNA polymerase, there's no way DNA polymerase can distinguish, oh, this is the wrong one. Yeah, it will pick up. Let's say this base is A, then this nucleotide, the dideoxynucleotide, will be added to the position of where the G is present on the template DNA. No, not G, T is present. Okay? So, but once it is incorporated, what do you think it will happen? The DNA synthesis will stop there. It can no longer proceed. Try to, would you kindly enough to examine this diagram? This is a growing chain. For now, you ignore this primer thing. I will explain this primer thing later today. But this will be the synthesizing growing chain. So this one nucleotide, the first one, second nucleotide, the third nucleotide, fourth nucleotide, and now this one is a fifth nucleotide is going to be added. Okay, so this fresh nucleotide is trying to be inserted into this chain, and the mechanism is always the growing chain will have, the last of the growing chain will have this, OH. Then because of that, this new nucleotide will be added to have this particular chemical reaction. So that there will be also another bond, so-called this phosphodiester bond. You don't have to remember the name. Will be established so that this nucleotide, now this 2-phosphate will be gone and single phosphate, the nucleotide having single phosphate will be linked. Okay. That's the mechanism of this DNA synthesis. But this one, this on the bottom figure, the last nucleotide was what? Was instead of a deoxynucleotide, this nucleotide was dideoxynucleotide, which is missing in this particular position, missing the oxygen. Without this oxygen, there's no new additional nucleotide that can be added. So it's just a stop there. That's the end. So that's why this technique is called dideoxy chain termination. So this whole scenario is this. You have regular whole bunch of regular nucleotide, but very rare, the unusual dideoxynucleotide is added. So it is always a chance event. You have to think that the template, when you try to do this synthesis, you don't provide only one single template, but template DNA is also very many. And also your nucleotide, deoxynucleotide is also many, and DNA polymerase is also many. So you are doing this synthesis reaction so many at the same time, so many repetitive DNA synthesis reaction is going on at the same time. At this scenario, if you have small amount of this dideoxynucleotide, this dideoxynucleotide will be inserted at random position of where it has corresponding the complementary base pair on according to the template nucleotide sequence. So in other words, point is, if you perform the DNA synthesis reaction in the test tube in this way, having many regular nucleotide, deoxynucleotide, having very small plus small amount of deoxynucleotide, then you will generate all the possible size of different size of DNA fragment. Their difference is only single nucleotide difference will be generated like this. Let's try to understand this concept first. Let's say this is original DNA sequence running from. If you want to do the polymerization, synthesis of this DNA sequence using this dideoxy chain termination method. So let's say 1, 2, 3, 4, 5, 6, all these different reactions at the same time going on. Why this fragment has only 3 nucleotides and then stop there? Because 1, 2, 3 at this position, G position, this is in color. This G in color represents the dideoxyguanine. Was unfortunately incorporated, and that's there. It ends. This fragment synthesis stopped there. On the other hand, another fragment have the fourth nucleotide, the A. At that position, this dideoxynucleotide has been inserted. So then its synthesis is stopped there. So in this way, all this, if you do this, if you carry out this synthetic reaction, all these different fragments having their difference in sizes by one nucleotide will be generated because this dideoxynucleotide will be inserted at random. Certain fragment, certain DNA reaction, unfortunately, get to pick up this dideoxynucleotide very early. Certain reaction, which is relatively fortunate, will be able to avoid picking up dideoxynucleotide very late, and then all the way down until the last minute, then bingo, you pick up this dideoxynucleotide and then end there. Another nucleotide synthesis get to pick up this dideoxynucleotide at this position so that it unfortunately couldn't finish all the way down, but simply finished there. So, mathematically speaking, if you carry out this reaction in the test tube, you will get all the possible different size of the DNA fragment because of this action of this dideoxynucleotide. That's the principle. So then your task is, anyhow you want to read all this DNA sequence information, all you gotta do is simply identify each what was the nucleotide that inserted at the end of this each different fragment, then you get the entire sequence of, hey, I don't want your help. Go away. Ask Siri. How can I? Okay. So is it not really clear enough? My explanation for this principle of dideoxy sequencing methods yet? You can still understand what is going on? To summarize, sum up, because of these chemical differences, so if you carry out this DNA sequence, DNA synthesis, then you are able to generate all different possible DNA fragments in different sizes. What I mean by different sizes is those different fragments will have the different size by single nucleotide because of this reason. Insertion of this dideoxynucleotides is random. Because if you provide all four different type of AGCT, different kind of dideoxynucleotide, and it's just a matter of at which position, let's say this is the original template. The DNA synthesis is simply try to insert complementary base pair. And then you can later interpret in terms of this complementarity rate. So that's how you read the DNA sequence. The previous diagram was a little more old-fashioned way because this figure illustrates this sequencing reaction using the fluorescence nucleotide. You can make later in these days you can make this nucleotide having a different color of a fluorescence dye. But in the days of when I was in the graduate school, there was no such thing like a fluorescent nucleotide. So you had to use actually the radioactive isotope labeled nucleotides. So in that case, there is no way you can distinguish whether that was A or G or C or T by your eyes. So you had to maintain this reaction in 4 different separate reactions, and in each of which you add different kind of like, this one is bugging me. Apple Intelligence. Keep interrupting me during while I'm speaking. This, all 4 different kind of dideoxynucleotide is added to each separate reaction chamber. In that way, then you can distinguish in which lane you had this different, which kind of dideoxynucleotide was added. So you know which was the last sequence that you can, depending on what type of dideoxynucleotide was added, you can sort them out in whether it is A or G or C or T, the last nucleotide. And as I explained in the last lecture, the very bottom in this type of gel electrophorese represent the smallest DNA fragment, and the second one is the second largest DNA fragment. So you can go all the way up to the largest fragment will be here. The smallest fragment will be at the bottom. So in this way, you can figure out the actual DNA sequence of this, your original DNA template. That's how you, actually I have done manually when I was in graduate school, did this. But in this age, nobody ever do this manually. All you gotta do is simply call to company and, hey, I'm going to send my DNA sample so that please get the DNA sequence for me. That's how the things change it. So actually, you don't really, even if you are a major in these days, you don't need to even, actually practically speaking, you don't need to understand this principle. Nobody will ask you, and without even knowing this, your life as a scientist, uh, presumed scientist, will never be jeopardized. However, still, using this principle actually will enrich your whole overall depth of the knowledge. That's why I'm introducing the actual principle behind this DNA sequencing. Although it may be very painful for majority of you to follow this principle, and who cares? Why do I have to know this? Yeah, I agree. But anyway, at least I tried. At least I tried to explain this principle so that actually this is kind of a in reverse way. You want to get the DNA sequence in order, but actually you get to figure out this DNA sequencing in backward. Try to synthesize, and during this synthesis, you deliberately create a scenario, situation where at each step you create somehow the failed DNA synthesis fragment. And then by identifying what was the last nucleotide in those failed fragments, you somehow figure out the sequence of this DNA fragment is the principle. So, okay, so that's and after getting this technology routine in every laboratory, now people thought about, hey, since we have this tool, now we can figure out all those DNA sequence of every different organisms. So how about, now is the time. The time is ripe for actually figuring out all those 3 billion, more than 3 billion pairs of the DNA nucleotide in our human genome was the project. So it started in 1990, okay, and officially ended in 2003, the Human Genome Project. So like before the Human Genome Project, of course, scientists first tried much smaller, simpler organisms DNA sequencing first. Like the first ever sequenced organism was the very small bacteria. Okay. Not to mention much simpler virus genome has been also identified, and then bacteria, and then some other simpler organism. And then they had they build up the confidence. Okay, now we think we can do that. Although this whole entire human genome consisting of more than 3 billion nucleotides. It's like, imagine that 3 billion nucleotides, if we consider each nucleotide as a single, like 12 font faces alphabet, and if you try to make a book having this human genome alphabet, it will, the page, number of page in regular book like A4 size page book, it will have about more than several million pages of the book. So your task is, you have never actually opened this book of several million pages, each page containing the regular, all those alphabets. Then you are for the first time opening, and then try to read from the first page 1 to up until the very last page, which is some million pages. Do you think that will be really easy? Well, really whole daunting task. Why do you want to do that? That's very crazy, stupid idea. Why do you want to read all your DNA sequence of the human? Why? Because that is the only way. Unless you read all those DNA sequence of human, there's no way you can actually know where is your gene. in which chromosome and which position, like those stuff. Without knowing those, you cannot move a single step forward in terms of, there's going to be a lot of occasions that you need to find the actual, the makeup of the gene. Like let's say, oh, you have some problem in the diabetes. So you suspect that the human insulin gene is something that responsible. Then you gotta actually know, you better know where is this human insulin genes located, in which chromosome, and what is the sequence is like. Without knowing, it's impossible to have any kind of progress toward such a thing. So it is identifying, like identifying all the genes on human. How many genes are there? Are there any genes that we haven't actually discovered yet in our genome? That's another very important curiosity. And where are they is also another very. So the purpose, the ultimate goal of this embarking human genome project is identify all the genes on our genome and also location. That's what we call some mapping. Mapping means identify all the genes and then arrange them in the proper order of a location of in each chromosome is what we call mapping the genome. Mapping the gene. So identifying the gene and mapping the genes was the goal of this human genome project, and it took something like more than 10 years. Okay, but actually, what is this? 2022? In reality, although officially, publicly, all those government, US government, it was like this project was initiated by US government, although several other countries also participated, including China and Japan, and all those European countries also participated in this project. So this is really a whole huge collaborative the effort, consortium effort from all those major laboratories all over the world. Mainly, of course, those main laboratories in the United States was the leading group, but also the foreign overseas laboratories also participated. And even with that, the actual real true completion year was like even only 2 years ago. Because it is not entirely completed because of this, mainly because of this pesky human Y chromosome. We couldn't really complete whole the sequence of the human Y chromosome because technical, the figuring out the entire correct accurate sequence of the Y chromosome technically was very, very challenging, so not able to. But those areas were not actually important area. No genes were there. So people ignored, okay, without those area, we still could technically consider it's all done. That's why in 2003 instead of 2022, the completion of the Human Genome Project was declared officially. Anyway, this genome, the characteristic of this human genome is actually a reference genome. What do you mean reference genome? Because the human, did you ever actually donate your sample for this human genome project? No, you didn't. And do you think your DNA sequence will be exactly identical to this human genome sequence that published out? No, because everybody's genome DNA sequence will be different. So actually, it's the whole this sequence was the like mixture of 8 representative of 8 different individuals of red blood cell samples. That's why we call that as a kind of a standard reference genome, as opposed to individual genome. Actually, after finishing this at like 2003, whatever, and then immediately the next step was identifying each different individual human genome. And guess who was the first person that whose genome was all identified? Can you guess? US president? That's who? James Watson. His genome was the first ever individual genome that was sequenced. So anyway, it was a huge, very expensive because it took more than 10 years. And the $3 billion was back, like the price, like 1990s price, $3 billion US dollar was a really, really huge, big chunk of the budget. But even with that, largely scientists believe that it was worth it. Even though, just like back then, my advisor actually asked me individually when I was in the US, hey, what do you think? Do you really think this human genome project is something that worth the thing? Don't you think it's a really stupid idea? Because then I asked him why, and then he was like, because even you finish this, everybody's individual DNA sequence will be different. And what's the point? How are you going to then figure out, like you spend this $3 billion for figuring out this one represented genome, and then how are you going to manage each individual DNA sequence, which is going to be all different? What's the point of this? Yeah, he had a point at the time. Because even if you have the standard DNA sequence of a human, you're individual. Actually, what matters to everybody, every different individual, is your actual DNA sequence, which is obviously different from this standard whatever. And how are you going to find out? Spending another 3 billion, waiting 10 years, and then you will be able to find. And hey, no, it's not. Okay? But he also, my advisor, my beloved, and with my utmost respect, my advisor, still didn't understand, couldn't envision that technology would change drastically later on. So that make this whole seemingly stupid task is actually doable and possible. But anyway, this human genome project was based upon conventional. Conventional means using that Sanger dideoxy chain termination reaction. The limitation of this dideoxy chain termination reaction is at single reaction you can read only something like a 600 to at most 1000 base pair nucleotide at a single time. So by that rate, if you want to complete this 3 billion nucleotide, you tell me how long it take to finish this thing. But anyway, this so-called, you don't really have to pay too much attention about this detailed technology, but the key point is it is very complex and it will take very long to finish, so-called this conventional. But there is no other alternative. Those scientists who involved in this project are all brilliant, almost near genius, but still, there was no other way around but simply had to stick with this very slow conventional way of figuring out. Basically, this so-called map-based genome sequencing is something like you have to figure out the actual order of a big chunk of the large fragment of DNA on each different chromosome. You have to get the order first, and then get the small fragment and then sequence, and then try to reassemble, reorder this thing is the basic things. However, something happened. So in 1998, it was like, meet this actual human genome project is going on. But they were supposed to finish, like they officially announced that in the beginning of 1990 that we should be able to finish this genome project around the end of this decade, something like so in the year 2000 around. So you should be able to do something like those kind of expectations, but it was moving very slow. course, once you start anything, you will encounter unexpected any difficulties, challenges. So they were kind of struggling. And then this gentleman, Craig Venter, is really a very weird, strange gentleman, like even much weirder than this guy, the, the Tesla CEO. Uh, this guy is much more strange guy, I believe. And he's an entrepreneur. He's a billionaire businessman, and he owned this company, Celera Genomics. Somehow, out of nowhere, in somewhere in 1998, he declared, "Hey, you guys, what the X you guys are doing? I think I can finish this whole genome project, or whatever it is, within 3 years. Within the next 3 years, I will do it." And everybody laughed at him. What? Hey, we are top notch scientists from all over the world. You are single only, pathetic single company, CEO, whatever. I don't care how much money you have, but we've been doing this for nearly 10 hours from all this leading laboratory in the United States all together, even getting the help from Europe and Japan and China. And you guys, what makes you think that you can beat us, and even within 3 years you can finish this thing? But he did. That scared everybody. So he declared, I can do this in 3 years, and then he kept his promise. Why? Because he knew something new methods should be introduced. That's what he believed. You guys doing this conventional way, so-called, you will never get. His new method is called shotgun sequencing, whatever. Here is the comparison between the conventional, the government method versus Craig Venter's method. By the way, this Craig Venter, not only did he accomplish this human genome project sequencing, whatever, in 3 years, but also he's the first one who ever created the first artificial cell. He actually made the artificial cell. So he maintained this created artificial cell in his place. The big difference is this whole thing, if your task was to get the identify whole DNA sequence of this entire chromosome, for example, his approach is get the random, chopped up, very small, tiny fragment in large quantities, in repetition. The government method is, first you generate some orderly fashionable, large enough fragment, and then you go from there to get the sequence, and then you reassemble this order out of this sequence. You're doing, of course, that is much more reasonable, understandable way of doing things. However, by doing this, it takes a long time. That's why it took even all those different laboratories participating together, it took 10 years and never finished yet. But this guy did this, and then once you generate the whole this, let computer figure this thing whole out, the order, is his strategy. Human can never be able to get all this right order. But hey, computers, why don't you come over here and then try to figure this whole thing out, was his approach. And then he did. He succeeded. So anyway, this is also another kind of a technical aspect, so you don't really have to pay too much attention. Sometimes some people try to show up his knowledge, his degree of knowledge, and then what's the coverage of, so to speak, what's the coverage of this DNA sequencing or anything like that? Don't be intimidated by such. There's nothing. Coverage means how much deep this repetition. The more you have repeated DNA sequencing, the more accurate your result will be, type of this relationship. So coverage is nothing. Anyway, as long as you have enough coverage of this thing, this shotgun DNA sequencing will work perfectly fine, is the lesson that we learned. Okay. Anyway, so looks like, so toward the end of that decade, like nearing the year 2000, like it turned out that actually the Celera is about to accomplish these things. And now everybody is in panic. Everybody who is involved in this, the government human genome project was like in panic. Why? Because it's not about their like personal ego or pride or anything like. But the biggest danger is what? Guess what? This Celera, this Craig Venter is a private businessman. So once he finished this DNA sequencing first, then all the sequencing information, he would put a patent on it and then keep out of touch of everybody. No one touches it but me. I have only access to this human genome information. So if you want to actually get access to this, pay me, type of thing. That would be really, really horrible disaster. So these government people and scientists really panicked. So now, this 2000, they simply, even without completing, they publicly published their result. Hey, this is partially finished our human genome, so that take the step in advance to secure what they have remain as a public domain property. So in order to prevent such the privatizing this precious information. Like similar thing, it was like this internet. Everybody enjoys all this internet technology in these days, but actually in the beginning, this internet technology was very exclusively to the US government, the US military usage. And only politically, somebody back there in US government, that's why that someone sometimes claimed that, hey, I was the one inventor of the internet, and then being laughed at by everybody. But anyway, he was actually right. He made it clear that this internet thing should be all like accessible to everybody in the whole world. So that's why in these days we are able to get this technology for free for everyone of us. Anyway, so they cut a deal. U.S. government intervened, and hey, whoa. So let's negotiate. So boss. This was back then Craig Venter at the year 2000, and everyone knows who this person is. And this was the head of the NIH, the US, Collins. And these two people, both of them were credited to have the human genome initiatives, the contributor. So all this credit were shared by Craig Venter as well as the US government effort. And so by doing so, this whole public access to this DNA sequencing information was saved. Without it, because probably we would have been living totally different type of a world in these days. Maybe next, the Wednesday's lecture, probably I can try to go over the actual meaning, the significance of this human genome information, especially in these days of a genome editing era. But anyway, so that's them. This NGS stands for Next Generation Genome Sequencing. So actually NGS, instead of NGS, it should have been NGGS probably, because there are two G's. But anyway, it's totally different, revolutionary technology compared with this conventional Sanger DNA sequencing method, which was the basic technique for the completion of the human genome technology. But in these days, nobody, unless in the small-scale laboratory DNA sequencing, still you use this conventional Sanger DNA sequencing. But if you want to actually do another genome scale sequencing, you never go back to. You only do this technology. This is a modern sequencing technology. Okay? So you don't use this so-called dideoxynucleotide anymore in this NGS. Hey, forget about this NGS. Totally new technology. But anyway, one keyword of this NGS is a high throughput. You can accomplish a huge amount of this job in very small time scale. That's what we call the high throughput. If you do so, then you can cut down the costs drastically. How? So in these days, actually. So remember, the initial original first human genome sequencing took more than 10 years, okay? And it cost more than $3 billion. Now, if you want to do same job, you can do it in one day. One day you're done. And everybody has $1,000 in your wallet. So everybody can get your own private individual DNA sequence done in one day. And this cost, $1,000 currently, is projected to be reduced to something like $30 or $40 within maybe next several years. Like. So, like I joked about everybody has $1,000 in your wallet, but it's not too much of a joke when I say, if I said everybody has about $30 or $40 in your wallet. Yeah. That's very affordable. Something like 3, 4만 원. Yeah, you can do it. That's why now the original question of why is it really worth it? Even if you have DNA sequence, human genome sequence, your DNA sequence will be different from this, and what's the point of doing this? Is that became moot point. Now every different individual can have their actual detailed DNA sequence information if you want to. Yeah.
그러면 내가 그 병원에서 나한테 그 네안데르탈 유래 그러한 이나 고혈압 유전자가 있는지 알아보려면 돈 얼마 들어요?
Even much less because all those sequence, like if you are interested in about certain specific genes that you have have a Neanderthal origin, then that task isn't even, you don't really have to go through the entire human genome sequence, but simply doing the technology, the PCR that we are going to take a look at later, not today maybe, then you can easily identify very cheaply, very quickly. Yeah, I don't really have to go over this type. This totally different type of a setup. Having, for example, your DNAs are trapped in this very small, tiny plate. All these different DNAs are trapped, and then somehow the sequence information can be obtained. Some of these, one of the such different techniques is being illustrated in this slide. Probably almost all of you is just meaningless. But anyway, the success of this NGS technology was actually thanks to two different, another very popular molecular technique called DNA microarray and the PCR. DNA microarray, of course, is also very widespread and important technology. So I wish it would be really good if I go over this technology in principle in the classroom, but no, I decided not to. Simply to give you some snapshot of what is it like, what is something DNA microarray. Your DNA fragment can be captured on the surface. All different kind of DNA sequence can be captured, fixed, so that this can serve as some kind of a reference point to identify all different type of physiological things, like including what kind of genes are expressed, your gene expression profile, and blah, 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 can be identified using this type of a setting. Using different type of a fluorescence image is basically what is called DNA microarray, or in many cases actually simply called a DNA chip, is what it is all about. So basically, keyword about this DNA chip is using this DNA chip, you can have information about your gene expression quickly. If, for example, if you are interested in, what is the difference between cancer cell and normal cells gene expression? Would there be any, in cancer cell, there are some kind of different genes are more particularly expressed compared with the normal cell? Type of thing can be easily identified, quickly, easily. That's why this so-called DNA chip or DNA microarray techniques is also very popular and widespread. And PCR is, name stands for polymerase chain reaction, likewise. Should I stop PCR and then try to go over some of your student question? What do you think? Or simply finish this PCR? Student question? Okay. So you get sick and tired of hearing the techniques too much today. So yeah, I agree. So I will stop today's lecture here and then try to go over, take care of your all the student question, because it's been being dragged, and so maybe in a sense it will be better for you to refresh your memory by going over this, your old questions. One question goes like this: is there any specific effect of a gene expression depending on the location, right? So you have a genes, and you have some enhancers and silencers. Do you still remember enhancer and silencers? Enhancers are the DNA sequences that to which the transcription activator protein bind. Silencers are what? Transcription repressors are bind the area. And especially for, Jesus, I'm getting too old. I should have catch. So especially this transcription activator is so essential for your gene expression. So, like, obviously, scientists became so interested about the enhancers, presence of these enhancers, because that's where all those the transcription activator proteins gathered to make certain gene active, right? And actually, to answer these questions, the location, as you saw the example in the lactase persistence, the phenomenon mutation, certain enhancers can be present in so far away from the actual start point of gene, but still can function without any problem. So these enhancers and silencers are relatively is unaffected by the relative location. It's regardless of relative location, this enhancers. That's another very distinct characteristics of this enhancers or silencers type of this regulatory element. And why do you think? Why, for example, enhancers, somehow, by what mechanism, enhancers can act, sometimes can act, provide their functions from such a far away location from the actual, the site of transcription? Guess what? Maybe should I give this as one of the final exam question? What do you think? So would you think about it? Why and how the enhancer can act as such a regulatory element from such a far away, remote location sometimes. Why? Why is a poor question? Probably how is the better question? How does enhancers act as such a regulatory element from such a remote, far away location? It's probably very legitimate, interesting type of question for your final exam. Okay, so get it. Let's get it. So you ask around, you ask to ChatGPT or whoever, and then all you gotta do is write down the answer. Oh yeah, this one. My bad is because, as I briefly mentioned the other day, or, actually enhance and promoter. The promoter is actually certain the term. Sometimes it's interchangeably used by the scientists. Like promoter, the actual correct narrow definition of promoter is the exact location, site to which what RNA polymerase, the enzyme RNA polymerase bind. However, generally, a lot of occasions, people, scientists, casually refer the entire regulatory area of the gene as a promoter region. So in that context, I use this promoter as a instead of enhancer. I should have been actually more carefully. I should have been more accurate, correct, precise, so that enhancer. I should have used enhancer, but I somehow I slipped my so I use the conventional casual way of referring them as a promoter. But yes, enhancer is more accurate. Allele partition into each gamete with the completion of meiosis. Yes, that's how they do allele. So like during meiosis, because I didn't have time to go over this way of sexual reproduction and cell division. So this is a good question. So when they have the cell division during the gamete production, like the eggs or sperm, this alleles, why the alleles are present? Because of this homologous chromosome. These two chromosomes are homologous, and each homologous pair now will be partitioned into two different dividing cells during the meiosis. One will be partitioned into this, and the other one will always be partitioned into the other cell. And that is actually, I forgot to mention, the most important event during meiosis. The most important event during meiosis is this pairing of homologous chromosome pairs. Why? Because by doing so, you can ensure that each different allele can be separated into different cells. Otherwise, it would have been much more difficult. Like if you didn't go through this like a pairing, identifying where is my partner homologous chromosome, and then let them meet just right before the cell division, then there will be higher chance of these two homologous chromosomes ending up in the same cell during this division. Then the result will be disaster as a gamete. If you have one extra allele, and that means the other cell is entirely missing that particular allele. You don't want to do that. Okay? You have to always make sure. The purpose of this meiosis is making sure that the product of this meiosis, each of these cells have exactly entire gene setup as a haploid now instead of diploid. You are diploid, but now you have to create haploid. But during the process, you should not lose any single gene. Just because you are trying to reduce your whole gene into half, then there is always a danger that you may lose some, like during this reduction. But by making sure this homologous pairing, you never miss such thing. Negative effects if only enhances are present on the DNA upstream. Not necessarily. And the other thing is, the reason why I put up this question is, actually, although generally all these enhancers are present on the DNA upstream usually, but certain enhancers are actually found the downstream of the gene. And even some very unusual enhancers are also found in introns too, if you still remember what the introns are. So they are really kind of a weird, strange things. Oh, yeah. First, what do you think? Why do you think we have a higher chance of catching cold if you left out cold for a long time? Your body temperature got lowered, okay? And what's got to do with your your immune strength? Your strength of the immune system is challenged by the colder temperature. That's why you have a higher risk of catching cold. And the other thing is this. Actually, I'm really sorry about this. So some of you understanding this English, yeah. I wish I could speak much more fluent and perfect English. That's my bad. But anyway, some of you, it will be difficult to follow this English lecture. So that if I could, I know actually some professors in this English lecture, but certain professors I know they run this system like operate in this way, like having some of the part emphasizing Korean, and some other professors even go over like in alternate class. Like one day you have an English lecture, and the other day Korea only lecture type of this mixed up alternating lecture too. I have heard. But in any case, actually, every at the end of every semester, I get this like evaluation sheet checking whether this lecture was actually conducted in 100% English or not, because it was supposed to. This lecture is supposed to go in 100% English. That's actually kind of my contract. So that's something that I should not break. And also, important point I thought about. At some point, I thought about how about if I actually have some of the part, at least some of the part, repeat in Korean so that have everybody has a happier, more understandable environment. But then, actually, in the past, several years ago, I tried actually. Then it getting nowhere because first that made me confused that which one is important, which one is not important is also difficult to decide. So by doing so, actually it dragged down the progress of this lecture. So that's so all I can say is I'm really sorry if you're having trouble in following this lecture, but all my kind of with all my sympathy, probably you could. Can you try to look over and review this the recorded version of this lecture? Try to catch up if you have a difficulty. Is or again, sorry, and already it's heading 15. I need more time actually. So I will. Once again, I should stop over here and then see you on Wednesday. Yeah.
그러니까
Mm-hmm.